with those who are interested in the central nervous system and how sound processing and ultimately speech come out of all of this. This is a very rich environment for this, and in case you didn't know it, Lars probably doesn't know this, this is one of the preeminent areas in the world, preeminent institutions in the world where this is occurring. So along with Princeton and the Max Planck Institute in Dresden, JITP is one of the three premier sites in which physics and biology have been commingled in a productive way. So it's a very special place for us to be. So what kind of things are we looking for at a higher level? This is the brain. The cerebral cortex is known to you all, cerebellum, brainstem, and whatnot. We know a lot about the visual brain. The visual brain is here towards the back of the brain. It has a number of areas that are defined by their architecture and other criteria. And we know something about what they do. So here's a stimulus. This is stolen from the SpaceX website. It's also an acoustic stimulus, but so we see all this activity, and what does the visual brain do with it? Well, we know that the first area of the visual brain, called V1, looks basically at the outlines of things. It does a sort of Picasso-like rendering of edges. So the spaceship itself, the rocket, the towers around it, the buildings, the edges of clouds, and so on, get defined there. Another higher area, V4, adds color to the scene. So the sky color, the ground color, and whatnot. Another area called MT is interested specifically in movement of the system. So each of these different modalities is represented at a different place in one of these areas. And there are other areas like V2, which represents stereoscopic depth. There's this geeky area, IT, the infratemporal area, that responds just to faces. So we know this much in the visual system and much more. We know much less in the auditory system. The auditory cortex is here. There's a primary cortex and then a belt of various surrounding areas. But we don't know that much about how they operate. We certainly don't know the detail that I just showed you. We know that there are higher areas that are very poorly defined, like Wernicke's area, that are involved in the apperception of speech sounds. So people who have a problem in this area, like a stroke, will have what's called a receptive aphasia. They can hear, but they can't make sense of speech. We know other areas, such as Broca's area, that are involved in productive aphasia. A person there, with a lesion there, can hear okay and recognize speech, but cannot produce it. So we really need to learn what the wiring is among these areas, how information flows to this region, how ultimately messages get over to Broca's area, and then from there down to the tongue, the larynx, and so on to produce speech. And there's some very productive work in these areas that we've learned about in the last few days at the presentations here. Another set of things we're doing is looking into how these remarkable structures, the hair bundles, are formed. They're remarkably regular, and they really look like they're stamped out by a mold or a cookie cutter of some sort. How can this be done? And it turns out that a physical model, due to Alan Turing, the great mathematician, can explain some of this. So this is the top of one of these cells, and you see the hexagonal pattern of the stereocilia, the single canocilium. If you suppose that there's some pink chemical coming from the canocilium and some of the green chemical coming from the boundary, you can make simple reactions and a prediction of how this would produce the pattern of a hair bundle. So you start out with this mess, nothing at all, but as soon as you turn on the reactions, something coalesces. And the something coalesces into neater and neater structures that look more and more like the hexagonal pattern of a hair bundle. So we're busy trying to understand now how this operates and also to demonstrate it experimentally. So with new microscopic techniques, it's possible to see hair bundles actually growing. So this is from a fish. This is a newly formed hair cell. This is a newly formed hair cell. As yet, they don't have hair bundles, unlike these more mature cells in which you can see the stereocilia sticking out. But in a time lapse, over about 12 hours, watch these stereocilia grow. So here they come. They're getting taller, taller, taller. And we hope eventually to be able to follow them all the way to full maturity. So we're trying to test these ideas of the chemical reactions that produce these mature hair bundles. The final thing I want to deal with is the problem of what happens as these cells deteriorate. What can be done, in a word, about hearing loss? We start out with these beautifully regular hair bundles. With age, with chemicals, with sound damage, they become disheveled. And the bad news is that in mammals, when one of these cells is damaged, it always dies. It is not replaced by cell division. So it's like the cells of one's brain or one's heart. Once they're gone for good. And because each of these cells or groups of cells represents a particular frequency of sound, those particular frequencies are lost as well. So one place we've investigated this is the lateral line organ of the zebrafish larva. So zebrafish are little aquarium fish that many of you have raised as kids. Their larvae are just a few millimeters in length at age 1 week. And along their side, they have something called the lateral line, these yellow organs which have been stained with a dye, each of which is a cluster of hair cells. These hair cells reach out into the water and they feel water motion. So they can feel water moving past the fish, they can feel the fish moving through the water, they can detect predators, they can detect prey. They're a very important sense for these fish. Each of these little clusters, which is called a neuromast, has hair cells, red and green in this case, red and blue, sorry, in this case, and they're of opposite polarity. The two red hair cells have their tall edge to your right. They're sensitive to stimuli, like the red arrow, in that right direction. The two blue cells are symmetrical and sensitive to stimuli in the opposite direction. These are produced in an interesting way. The ear sits just behind the eye, but from the ear, a cluster of cells called a primordium streaks off down the length of the animal when it's 2 or 3 days old. And as it goes, this primordium deposits clusters of cells. So here's the primordium chugging along, and a cluster of cells being dropped. And here's a quick time-lapse movie showing that. It's like a bus letting off passengers periodically and always dropping off about the same number. So the primordium is here. This is part of the tail of the fish. Here's one cluster of cells, a second cluster of cells, a third cluster of cells dropped off by this primordium, and finally it gets down to the tail maybe a day or so later. Now if we look at one of these clusters, we can see the hair cells developing within it. So to start, I want to point out this cell here, which is black at the outset, like these other black cells around. These cells that are fluorescent yellow or green are hair cells. There are in fact 1, 2, 3 pairs of hair cells here, 4 pairs of hair cells over here. This cell is turning green. It's decided it would like to be a hair cell, or more likely, it's going to be 2 hair cells. In just a minute, we'll see a very excited macrophage come. There it is. And this cell will now undergo a division. Pop into 2. Watch this cell. It's going to commit suicide. So it's gotten old and died. So there's sort of a conveyor belt system. New cells are formed at the North Pole and the South Pole. As they get older, they move to the center and eventually die and are replaced. Here's a schematic diagram of that. They're stem cells that are constantly making new cells, and they're always made in pairs. And interestingly, the pairs are always of the opposite orientation. So one cell, the one on your right, is sensitive to motion to your left. The cell on your left is sensitive to motion on your right. These cells then in pairs move down towards the equator from both directions and then finally age and die. The reason I bring this up is that we can now use this system to try to decode the molecular and genetic pathways that are involved in this progression. What is the stem cell? What is this amplifying cell? And what are the series of reactions involved in the progression to new hair cells? By decoding that, we hope to be able to learn how to evoke something similar in the mammal, including the human. One other thing about this system that's turned out to be interesting and instructive is, as I mentioned, the cells have 2 different orientations. Some oriented towards the fish's front, some oriented towards its rear. Those different cells have different nerve fibers because they represent different things. One of them represents movement of water towards the fish's back, one towards the fish's front. So a single nerve fiber, such as the one marked in red here, comes in and spreads over these cells and ends up at 4 days innervating just these three. You can see the little red spots, which are the synaptic contacts. And those three cells are all facing to your right. In other words, they're sensitive to motion to the right. This nerve fiber does not innervate the cells facing in the opposite direction. And in fact, there's another unmarked nerve fiber that does. So one wonders how the connection between these two is set up. And when we studied this, we found a great surprise. The supposition has always been that a hair cell just sits there waiting for a nerve fiber, here shown in magenta, to come along and make a contact with it. But what we found is that when newly formed hair cells are made, they participate actively in finding a nerve fiber. So when I play this time-lapse movie, you'll see this hair cell in particular put out a tentacle like an octopus, reach up and grab a nerve fiber. So here we go. Here comes the tentacle. It missed. Here comes the tentacle again. Now it's got the nerve fiber and reels it in. This is something totally unanticipated. Nobody's ever made movies of this before. And it gives us new clues to look for the molecules that are involved in this interaction. The other system we and others are working on is the mammalian system, in particular mice. Now, in animals such as the zebrafish that I just showed you, if the hair cells die, there are left over so-called supporting cells that are normally between the hair cells. And in zebrafish, and indeed in reptiles and birds, these supporting cells will undergo mitosis. They'll split, making new supporting cells. And some of those supporting cells, say these two, will then turn into new hair cells. And we'll be back where we started. This animal will be able to hear again. We can't do that. In mammals, what happens is that the supporting cells do not undergo proliferation. Instead, there can be some that turn into hair cells, but there are now too few supporting cells, and the whole thing tends to die. And this is what we're left with, just the scar where the hair cells once were. So we're trying to find out how to deal with that using the utricle, which is another organ from the mouse's ear. Here's the cochlea. The utricle, which is easier to dissect, it's nice and flat, has on the order of 2,000 or 3,000 to 10,000 hair cells, shown here in this nice pink color. It's the organ, remember, that's sensitive to up and down and particularly side-to-side motions of the body, but the hair cells are very similar. And what we've done in this case is look at the capability of this utricle to make new supporting cells and new hair cells. So this white label, each of these white spots, represents a hair cell that's dividing. And what we find is at P0, which is the day of birth in a mouse, there are a lot of cells that are still capable of dividing. But just over a week later, there are only a few left. And this is a graph showing the same thing. Just after birth, the capacity to divide diminishes enormously, and for the rest of the animal's life, no new supporting cells, no new hair cells can be made. So we ask what genes are changing around that time. There are a number that were already known for various reasons, but we discovered this red group of genes called SOX-C changes abruptly. Their expression of each of these genes falls, except this one, which is a contrary gene, goes in the opposite direction. And this is very exciting because we can manipulate such genes. We can use a virus, for example, to re-inject these genes and turn them on. So here we have a cell, a utricle that's a couple of weeks old. There are no cell divisions at all in its interior anymore. But if we use a virus to inject this gene for SOX4 or the gene for SOX11, the particular molecules I mentioned, we turn things back on. So this is really a hopeful thing because it means we may be able to find ways of reinitiating the proliferation of cells and then the differentiation of new hair cells. Now, we don't want to do that with virus. None of us much want viruses to be injected. But what one can do is to look for small molecules, that is drugs, that do the same thing. And our group has screened 65,000 small molecules and found a handful of them that have the right sort of effects. So we'll try these first in this preparation in a dish, and if any of those work out, then we'll try it in a real mouse and see what happens. So the last thing I want to mention is what happens until these strategies work. The answer is the cochlear prosthesis, the cochlear implant. And you already can understand it works Because in the normal cochlea, as I've mentioned, a particular tone evokes motion of a particular group of hair cells. That activates a particular group of nerve fibers. If every cell has been effaced, one can nonetheless signal the brain that a particular note is being heard if one could use a little electrode like this red wire to shock just those nerve fibers.